My name is David Scherr. I'm a PhD student in the Biological Systems Engineering Department. So originally, I'm from Plainsboro, New Jersey, a little town outside of Princeton. I got my undergraduate degree in biochemistry and chemistry from the University of Miami, the one in Florida, not the one in Ohio, the fun one. And then after that, I immediately decided to get my PhD, and I joined the Biological Systems Engineering Department in the fall of 2017, and I've been there since. And I'm set to graduate next year, and knock on wood, hope everything goes well. Let's see what happens. So a little bit more about me. I'm a big basketball fan, so that's me at a Charlotte Hornets game about 5 or 6 months ago. I'm also big into the outdoors. So it's me and my dad before we're about to go on a rafting trip. And then the last picture is at Sequoia National Park, where the sequoia trees and the redwood trees are, the biggest trees in the whole world. They're awesome. So that's my arms, outstretched in front of a sequoia tree. Huge, massive things. Highly recommend it if you ever make it out to California. Beautiful national parks. Absolutely beautiful. And so like I said, I'm in the Biological Systems Engineering Department, and a little bit about that. So we are a unique department in that we are, I believe, the only department that is both in the College of Engineering and the College of Ag and Life Sciences. And so we have this cool crossover. And our research really specializes in two distinct areas. So the first one is water resources and water quality and water management. So that's kind of, looking at watershed, looking for water pollution. In fact, you guys, I believe, were with Hannah earlier, who's also in BSc. And Hannah just did some really great research on looking at water quality for West Virginia wells. So that's her side of things. I'm on the other side of the department, which is bioprocessing. So that is looking at using organic life to make chemicals for us, to make energy for us, to make pharmaceuticals, all that stuff, either through genetic manipulation or just good engineering practices. So my real passion in research is in metabolism, which is how an organism turns one chemical into another chemical. And this happens in every source of life everywhere. And as an example of that, this is the TCA cycle, or citric acid cycle, which you probably saw in biology. And this is how your life takes, how your body will take acetic acid and basically break it down into carbon dioxide while extracting a bunch of electrons and a bunch of energy from it. So the reason metabolic engineering is very important is it's also called genetic engineering. And what I'm interested in is seeing if processes that are normally done in a large-scale industrial power plant could be done inside of a cell. So for example, let's say we have a factory meant to make plastic out of oil. Well, instead of turning oil to plastic, we could feed a genetically modified bacteria sugar, and that bacteria will turn sugar into a biodegradable plastic that is safer for the environment and could potentially cost less than if it was made by oil. And that's where I come in. I'm trying to help develop these processes to eliminate oil so we can make our chemicals through renewable sources. And there are tons of jobs all over the sector in these fields. This is really biotechnology, which is the fastest growing field of just industrial jobs out there right now. So a couple examples, Johnson & Johnson, they make pharmaceuticals, very important, very good stuff there. Beyond Meat, so all the fake meat companies, all those fake meat products require a ton of chemicals and a ton of good processing to go in them to make them. Monsanto, huge in American and foreign agriculture. Both ExxonMobil and Alganol Biofuels are looking at trying to make fuel for your car from algae. So I wanted to include them too. I really like so Alganol is based in Fort Myers, Florida. So if you guys want to work in Florida, it's a good job. Really warm there, very nice weather. And then finally, another favorite company of mine is Carbon Biotech. That's looking at ways to capture carbon dioxide in our air and convert it into some kind of product. Ideally like a plastic. Like imagine if we could suck all the carbon out of the air and turn it into like lids for a cup or something like that. I think that'd be pretty cool. So for this activity, like I said, we are going to be using the pipe cleaners that you guys should have gotten in your supply. So we should have 3 different colors, blue, orange, and yellow. Unfortunately, I don't have the yellow ones. I got white. So you're going to have to pretend that my whites are your yellows, and we'll go from there. So, sorry, I went ahead of myself. So right now, I'd like to take 2 minutes, get a pair of scissors, and cut all your pipe cleaners in half. So it should have 6 total. It should be 2 of each color. You're cutting each in half. So then you're going to have 12 total pipe cleaners. So get some scissors. I'll give you guys, 5-ish minutes to find scissors and just cut your pipe cleaners in half. I recommend doing two at a time. If you do 3, it gets a little bit messy. There we go. Cut your pipe cleaners. All right. So after they're cut, you want to separate them into 2 groups. Each group will have 6 pipe cleaners, 2 of each color. So you're going to have, this is one group. I have 2 of each color. Then I'm going to have another group. And we're going to turn our pipe cleaners into proteins. And you're going to learn about protein function, a little bit about, not protein function. You're going to learn about how proteins fold with these pipe cleaners, which is a very important thing for a lot of reasons. And you'll find out why. Don't want to review everything. All right, got my 2 groups. I'm going to wait a little bit. All right, so one last time, once they're cut in half, you want to separate them into 2 groups, each with 6 pipe cleaners, 2 of each color. And these will be turned into 2 different proteins, which we'll get into. So while you're cutting, if you're still not doing that, let's answer some pre-activity questions. So if you don't want to unmute yourself, that's fine. We can use the chat box, but I want to get a good idea of what you guys already know. That'll kind of help me with how much information I should give you going forward. So with that in mind, when you think of, sorry, I got to move this chat box. It's in the way of everything. So when you hear the word protein, like what is the first thing that pops in your head? Just that first thought. Then you can put that in the chat box. Energy, very good. Proteins are very important for energy. They take energy to make, and they help catalyze reactions, costing us less energy in the end. What else do proteins do? Meat and fish. Yep. Meat and fish, full of protein. And chicken, too. Chicken's my favorite of all meats. If I had to pick one, I'm a big chicken guy. Anybody else think of proteins? Okay, so what's something that you already know about proteins? Like, just give me like just a random fact or just a random thing you kind of know about them. I'm just curious. They give you energy. That's true. Build muscle, very good. If you want to build muscle, you got to eat a lot of protein to build it up. Anybody else got one? All right, then we'll move on to the last question. So what are some things you don't understand about proteins? They are made of amino acids. Very good. We'll get into that in a little bit as well. But what are some things about them that you don't understand that you hear, maybe it was some news article about proteins. You're like, what? That makes no sense. Just, I don't know, or something you're curious about, just something about proteins which you want to learn more about. That's a thing. Does anybody have anything like that? Alrighty, well, we're just gonna move right along then. So we can start this good video. Their relation to carbohydrates. Ooh, I like that. They are related. And maybe I'll get into that a little bit. If I don't get into that, you can ask me about that at the end of my lecture. More about the shape. Well, we are doing protein folding, so we're going to do a lot about their shape. And here is the video.
Protein is a building block of life, just like you hear on a Weet-Bix ad or something. And these building blocks make up all of the bits that go into building our cells and our bodies. So proteins do very, very many different things. They can make up part of your muscle, or they can make up part of how you grow your fingernails, or they can have much smaller, more specified jobs inside of cells in your body. Some proteins are needed to send messages across cells from one side to the other, or to the next cell maybe. And some proteins are needed to organise the bits and pieces that are inside your cell. Inside your cell you have protein-making factories called ribosomes. These themselves are made of proteins, and they make all the different proteins that you need for all the processes in your body. Some proteins are enzymes that need to break down energy to give you the ability to move and to think. And some proteins form part of the cell membrane, or the packaging that goes around the cell that connects it with other cells around it. And some proteins are involved in organising your DNA or your chromosomes to make sure that they are doing the right things at the right times in the cell, and that when the cell divides that they don't get tangled and scrambled together. And also that the membrane around the DNA, so you have the DNA is in what's called a nucleus, and the nucleus has a membrane around it. It controls what goes in and out of the nucleus, and that's another job for proteins as well. So you name anything that your body does and there'll be proteins involved in doing that, and they come in all different shapes and all different sizes. So some of the most famous proteins are things like hemoglobin, and hemoglobin carries your oxygen around in your body and in your blood, and hemoglobin is the particular colour it is, red, because of how it carries oxygen. And oxygenated hemoglobin has a particular red colour, and deoxygenated hemoglobin is a more browny colour. So our blood is red because of the properties of hemoglobin, a protein in our bodies.
All right, I really enjoy that video. Learn about their shape. Well, that's what the focus is. So, just to briefly go over what the doctor mentioned. So proteins do a ton of different things, which is why I'm so interested in them. They build up your tissue, get you big and strong. They will catalyze reactions, which is what I'm interested in, as they will turn one chemical into another chemical. They help with cell structure. Like if you ever heard of like a cell is like a microskeleton or cytoskeleton, that's proteins just linked, kind of linked together in a large chain. They're a source of energy, as some of you have mentioned earlier, so you know that. And they also will transport a certain chemical from one part of the cell to another. So very important for that as well. So proteins are made up of 20 amino acids, and you don't have to know their names, know anything about them really. In fact, I didn't have to memorize, well, I did have to memorize all the amino acids, but not until I was a senior in college. So you have plenty of time to really get this down. But what I want you to focus on is that there are three main types of amino acids, even though there's four shown here. There's nonpolar or hydrophobic, which means that they don't interact with water. In fact, they are repelled and repulsed by water. The other two are positively charged and negatively charged. And those like water. They want to be around water and form the hydrogen bonds. And then polar can be positively or negatively charged, but that depends on what else is going on in the environment. So that's a little bit more of a complex question, which we're not going to get into because you don't need to know that. What you do need to know is how proteins make a bond. And they make something called a peptide bond. So every protein has a nitrogen group and a carboxylic acid group. So one hydrogen from the nitrogen group will react with the oxygen and hydrogen of the carboxylic acid, and they will form water. And as they form water, a bond is made between the carbon and the nitrogen, known as a peptide bond. And these are strong. They will not break. Very strong bonds. The other kind of bond and a protein will form is a hydrogen bond, which only happens between the positive and the negatively charged amino acids. So now we're going to get into folding these suckers. So our yellow, which is my white, is going to be the nonpolar hydrophobic. Blue is negatively charged, and orange is positively charged. And now we're going to make our peptide bonds. So how's that going to look? Well, I hope that on the big screen right now, I don't know if mine's the biggest, but basically you make an X with them and you just fold them over each other, just like that. And a couple folds, bam, peptide bond. Not too hard, right? So I would like it if you could put together your proteins in any particular order. Just take your 6 amino acids and fold them together, or make your peptide bonds. And then you will have 2 completed proteins. I'm going to do it now. I hope you follow along with me. All right, I got my first protein done. Ta-da! Beautiful. Now it's time to get the second one going. Don't worry if I'm going a little bit faster than you. I've done this like 20 times. Making my second protein. Also, if you have any questions about any of this in general, feel free to put it in the chat, and I will answer it to the best of my ability. Also, if I'm going too fast, don't feel. Please slow me down, because I have rehearsed this lecture and done it a couple times now, so I might be going kind of fast because I'm used to it. So if I am, please tell me I'm going too fast. All right, one more. And my 2 proteins are done. Number 1 and number 2. I'm going to give you guys another couple minutes to finish your own. You can hold them up if you want when you're done, so I know you're good to go. Some beautiful, beautiful proteins. And don't feel bad if they look ugly or something. You're going to be folding them and unfolding them a couple times, so that's okay. The important thing is just the concept behind it all, I guess. Oh, I dropped mine. All right, everyone appears to be good, so we're going to get the show back on the road. Okay, so you got your 2 proteins. So what you just, all right, so now we're going to really fold them into a protein shape, which is a glob, pretty much. Proteins are globular in nature. And these globs are kind of guided by principles in chemistry. And one of the main principles is the hydrophobic effect. So like I said, your yellow amino acids are nonpolar. They're hydrophobic. They don't want to touch water at all. They are repulsed by water. It just disgusts them. So what they're going to do is they're going to force themselves into the middle of your protein so as to not touch the water to the best of their ability. While your positively charged and negatively charged amino acids, they like water. They want to be surrounded by it. So they're going to push out. So you see here how the green side chains are nonpolar and they are pushed into the middle, while the polar, the positively charged and negatively charged side chains are pushed out because they want to touch the water. So we're going to fold our proteins using that principle. So we're pushing the nonpolar sections inward, and the polar outward. So what does that look like? Well, once again, you're going to be looking for a glob shape. So here's mine. I got my two non-polars are at my end. So I'm going to pretend that my finger is like right in the middle. I'm going to push them right into the middle, hold them onto each other, then surround them with my hydrophilic positive charge and negatively charged ones. And voila, we have a protein, and it's very globby. So I got a lot of the white is tucked in, or I guess your yellow will be tucked into the middle with your blue and orange residues on the outside. Ta-da! Ain't it pretty? So I'm going to do that again with my second one. If you want to watch me fold them again. That looks great, Gracie. So once again, push it into the middle of my invisible middle. Surrounded by the hydrophobic and hydrophilic, surrounded by the positively charged and negatively charged amino acids. Wrap it around, maybe crush it in my hand, make it very bally, and voila, another protein. So once again, it does not have to look perfect, just has to be a globby shape, and you want your hydrophobic guys in the middle. And that is a realistic protein, even though it looks kind of messy. I'll give you guys another minute or so to make your globs. So what you just made was technically the secondary structure of a protein. So I'm going to go slow here and break it down. So when you did your the first time you made your peptide bonds, what you made was the primary protein structure, which is just the amino acids and the peptide bonds in a straight line. Afterwards, the hydrophobic effect starts taking place and we get some folding. This leads to the secondary structure, which is an alpha helix or a beta sheet. Whether it's alpha helix or beta sheet depends on where the hydrogen bonds take place. The hydrogen bonds can either be parallel or anti-parallel, making a beta sheet, or they can be kind of up and down, kind of like your single strand of DNA, giving you the alpha helix. So one protein can have multiple alpha helixes and multiple beta sheets. A protein can be like 500 amino acids. These things are huge. So because of that, an alpha helix and a beta sheet can then interact with each other on the same protein, giving you the tertiary structure. And then finally, multiple tertiary structure proteins can come together to form a quaternary structure. So by that I mean, from the video earlier, hemoglobin is a quaternary structure protein. It's made of 4 identical subunits of tertiary proteins coming together to make our hemoglobin, which helps keep the iron in the middle, so then the oxygen can bond, and then it goes up the blood and down the blood and all that fun stuff. But we're really only focused on the secondary structure, and this is only like we can't make an alpha helix with 6 amino acids. It's just not enough. But I just wanted to, this is more of just an example of kind of the folding processes. Now, just as a protein can fold, it can also unfold. When a protein unfolds, that's called denaturing. And denaturing can happen for a lot of reasons. So who here has cooked a steak before or cooked steak? So if you go to a restaurant and you get a burger or a steak, you can order it rare, medium, or well done, or in between, right? And the longer the steak cooks, the more the middle of it goes from a reddish color to a gray color. Well, as you guys said before, meat is full of protein. So what's happening is as you cook the burger or steak for longer, the proteins in that steak are denaturing. And when they denature, that's where that gray color starts to appear. while undenatured natural proteins give meat that kind of red color for when they're not totally denatured. So that's just one way that protein structure is kind of in your food already, whether you knew it or not. So proteins can denature for a lot of different reasons. One is new chemicals in their environment. So this is guanidine hydrochloride, and that is urea. And these kind of upset the hydrogen bonds inside of a protein, and it will break down. Another is changes in pH, whether it's very acidic or very basic. That'll also cause the protein to lose its structure and break down and denature. And the final one, using the example of a steak or burger again, is heat. You can heat up a protein, and that'll cause it to unfold. Oh no! You drop your protein into a vat of boiling water. It's gonna unfold. So take one of your proteins, and let's make it into a straight line again. It got unfolded. Straight line. Now, I want you to try something a little hard. I want to see if you guys can refold your protein exactly the same way that you did before. Try to refold it to look exactly the same as it did before. So I don't know about you, but my thing looks a little different than it did before. Was anybody able to fold it back the same way? Just a head nod, head no, no, no. I'm getting a lot of no's. Well, that's okay, because that is the key principle I wanted to show. It's very hard to fold the protein the same way twice. In fact, your body has developed ways to get your protein to fold exactly the same way every single time. And these are called chaperones. I like to imagine how a chaperone at some field trip with like little 5 or 8-year-old kids. The kids are always like running away. The chaperone is like, no, come back, stay, be organized. That's kind of what these proteins do. They want the protein to fold in one particular manner. So here, for example, is a chaperone called trick. And when a certain protein goes inside trick, the structure of the trick chaperone is very specific, forcing the protein to only take one possible shape. And thus, the protein is folded exactly the same way every time. So let's do some brainstorming. So what are some things you could think of, like a way to get our pipe cleaners to fold exactly the same every time? If you can think of anything, just put it in the chat or say it out loud. Like for example, one thing I thought of is maybe like a big box, like or not a box, but like a cylinder, which you kind of shove the protein down into. As you shove it down with force, it always makes the same shape. Another idea could be like super glue, but I feel like super glue is kind of like cheating. I don't know, it seems a little bit too easy. Can you guys think of any ideas? A stapler, maybe? The last group I did, one of the girls suggested a cookie cutter. Or not a cookie cutter, but yeah, a cookie cutter mold. Because those molds, you could force it the same shape every single time. I thought that was a very cool idea. Can anybody think of anything? Alrighty, that's fine. We'll just continue on. All right, so this is our last time unfolding the proteins, and this is to demonstrate something called amyloid formation, which is very important. So now I want you to unfold half of each of your proteins. So have, 3-ish amino acids available. Sorry, one of mine broke. And then combine them. Try to refold them together to form one protein out of your original 2. I'm going to try that now. And oh man, this is messy. Yeesh. So this is my mega protein that got formed. Not very clean looking, if you ask me. So, do you think your new protein is functional? Do you think it would work? Seen a couple head shakes. And yeah, you are right. A new protein, protein function is very specific. So when it bonds to another protein, that really eliminates its function, which is bad. And on top of that, a lot more bad things could happen. And we're going to talk about that bad thing now. So here's your normally folded protein, okay? So something could go wrong in the environment that causes the protein to be misfolded, which means it's not going to function. It's just a misfolded protein. And the name for those are amyloids. Now, amyloids have a very bad habit of sticking next to each other. And they'll keep building and building and building and building on top of each other until you get a whole fiber made of misfolded protein. Now, this fiber is like plaque for your teeth in that it just gets stuck in places. And it's really bad for your body. The main disease caused by amyloid formation is Alzheimer's. And I'm sure most of you have heard of Alzheimer's or know a little bit about it. It's a brain disease where you lose your memories and you lose your ability to function as an individual. And the way it works is that over years and years and years, these amyloids will build up inside of your brain like plaque on your teeth, and they will stick to the neurons in your brain, preventing them from talking to each other. So Alzheimer's is a protein folding disease. And it's not the only one. Mad cow disease is also from misfolded proteins. So when proteins aren't folded right, a lot of bad things could happen. And a lot of scientists now are looking at amyloid formation to figure out if there's a chemical or some kind of additive which we could put in someone's brain, which will break apart this plaque without hurting the person. And unfortunately, that's very difficult, and we haven't figured it out yet. I'm sure there are some clinical trials that have proven some progress, but there's no cure for Alzheimer's. I mean, I haven't heard of one. So this is still a very big problem, and there's tons of research going to how can we reverse this bad folding of a protein. And that's kind of the big takeaway that I want you to get, is that protein folding is linked to our health, and understanding how they fold can lead to better medicine and better design down the road. So now that we went through our activity and you made your pipe cleaners, give me something new that you learned. I'm sure you guys learned a couple new things, so put that in the chat or say it out loud, whatever makes you comfortable.
I didn't know that Alzheimer's was because of the bad folding. I just thought it was, I didn't think it had with proteins.
Yeah, that's really why I wanted to, have that kind of be the focal point of my lesson, but I wanted to kind of lead you there for that build up, I guess, because it's very important and something that a lot of people don't realize. So I'm glad that you took that away. Does anybody else have something?
I agree with Gracie. I didn't know that either, and I thought that was really interesting.
Well, I'm glad I could teach you that. Do you view proteins? Do you view proteins in a different light now because of this, or anything new that you learned? I guess that's the first question. Okay, and I guess last, what are your biggest takeaways from this lecture, if you have any big takeaways?
Probably that proteins and protein folding are important to our health and like our mental health too.
Yeah, they do impact your mental health. I guess one thing I could include, if you're very interested about this, is that there's a type of unfolded protein called prions, Prion, and prion disease research is very big as well. Prions are different than amyloids because they don't form a plaque, but what they can do is a prion can bump into a properly folded protein and cause it to misfold, which is bad. And it kind of sets off a chain reaction of more proteins unfolding. So protein folding and disease very linked. Any other takeaways? Okay, well, I'll finish up this lecture with my other, as did my previous lectures, where you can now ask me questions. So if you want an idea, I did science as my undergrad. I'm now in a engineering grad program. I've been tons different to different colleges up and down the East Coast. So any questions you have about that kind of stuff, I am more than happy to help you out. So any of that will be great now if you have anything.
Did you like University of Miami?
Yeah, I did like it. I had a pretty good time. Haven't visited in a while because most of my friends have graduated now, but I really enjoyed my time there.
That's cool.
You said you were from New Jersey and you went to Miami. How did you choose Miami?
Well, honestly, I chose Miami because they gave me a very big scholarship. They were my second cheapest school behind Rutgers, which is a public school in New Jersey. And I visited Rutgers and did not like it at all, really. It's just like Rutgers had this policy, which, by the way, if other schools have this, I'd say it's a very questionable policy, in my opinion, of the engineering students were isolated from the rest of campus, as in their classrooms were separate, and I would have been living in a dorm with only other engineers, which I was not excited for. So that was a big reason. But yeah, so Miami gave me a good scholarship. It was between them and Tulane in New Orleans. And Miami gave me a better deal, so I went with that. Any other questions?
What types of classes in high school did you take to set you up for your college career?
So the big three would be AP Bio, AP Chem, and AP Calc. And I really like those classes too. I took the AP tests for all of them, did pretty good there. So I recommend those. Besides that, I think statistics is greatly underappreciated. And not every high school offers a stat class, but if they do, I recommend it. It's also really useful. One thing I found out, which I wish I was taught in high school and in college as well, is that it's very easy to lie with numbers. So statistics will help you with your own critical thinking when viewing other people's stat work, which I found has been helpful. Any other questions? All right, well, you guys have been great. Thank you for your time. I really appreciate you coming out, and I hope that you learned something important that you'll carry with you. So have a good rest of your day, and thank you for listening to my presentation.
Thank you.
Goodbye, everybody.
Thank you.
You're welcome.